Good afternoon. Can I just check if the presenter has joined us today? Just give us a few more minutes, yeah?
Okay. Yeah.
Still waiting for the speaker to join.
Right. Yeah. We have been in contact with the speaker. We wanted to see, yeah, whether the speaker has no, call it join in later.
Okay. Just bear with us. All right, to all participants, just bear with us. I've been told that the speaker will join us in 5 minutes, yeah? Okay, the speaker is from Germany, therefore we anticipate some time difference.
Sampai ni dia lebih tinggi lah. Okay. Sama juga kalau kita pakai dekat ankle, kita akan elevate ankle tu. Okay. Right. Okay. After that 20 minutes, kita akan remove. So usually remove, dia ada dua cara. Sama ada kita remove dulu air tu ataupun kita remove dulu alat yang kita pakai, appliance yang kita pakai tu. Tapi usually dekat sini kita remove
I think, Dr. Tasneem, are you there? Are you with us, Dr. Tasneem?
Yes, can you hear me?
Okay, hold on.
Yeah, nice to see you, Dr. Tasneem.
Thank you. Sorry, actually, there was a change of time this weekend. So it's 8 o'clock. So
Okay.
Yeah, the
uh,
daylight differences. Yeah, we understand.
yeah,
Yeah,
yeah. Sorry. So this
no
was
worries. No worries.
fortunately I was almost ready. I just sorry for this. I
Perfect.
think
Yeah, that's perfect.
All right.
Yeah. All right.
So a very
okay.
good afternoon to
Good
all
afternoon.
professors, lecturers, staff, students, and all participants, and a very good day to you, Dr. Tasneem. I believe it's 8 a.m. now in Germany, right?
Yeah,
So
yeah.
to all, welcome to our IMMB webinar series 7 for this year 2021. So before I start, I will just introduce the speaker to us. Dr. Tasneem Khanam is a senior postdoc at University Clinic Muenster, Germany, with the Department of Pediatric Hematology and Oncology. She is an active member of the European International Group for Childhood Non-Hodgkin Lymphoma. She obtained her Bachelor of Science and Master of Science in Microbiology from the University of Bangalore, India, and her PhD from PhD Institute for Experimental Pathology, University of Muenster, Germany.
Yeah.
She has published numerous publications, granted many awards and prizes for the work that she has done. So I won't take too much time. Dr. Tasneem, over to you with your presentation.
Yeah. Okay. I need to share the screen. So normally your a window, your entire screen. One moment. How I should. I'm so used to the Zoom,
You see
and I
the
have
box with the arrow, and then you just right at the bottom.
yes, and then your a window, right? I have to choose. Okay.
Right.
Oh, okay. 1 minute. I just need to. Yeah. Okay. Just a minute. Since I was not ready, it's a little bit of confusion.
Right.
Yeah.
Yep, we can see your
So
slide.
can you?
Yeah. All right, perfect.
Okay. So last 5 years, I'm working in the Department of Pediatric Hematology Oncology in University Clinic Muenster. And my work was mainly to characterize at molecular level T lymphoblastic lymphoma. Our group is working very actively in non-Hodgkin lymphoma field to improve the treatment outcomes, and also they're involved in various clinical trials and so on. And I was mainly focused on working on molecular characterization of T lymphoblastic lymphoma. So we used an integrated approach where we used combined methods to identify molecular markers of clinical significance. So the main work was published recently in Blood, and I'm going to present this project today. My talk will focus on the following points. I will briefly introduce the state of the art when I started the project. The first part will be more about genome-wide screening for recurrent genetic and epigenetic alterations in T lymphoblastic lymphoma, designing of a targeted panel for T lymphoblastic lymphoma. Amongst the results, I'm going to briefly talk about schematic display of localization and frequencies of mutations for candidate genes, their prognostic relevance, and modeling of some of the mutations in silico for one of the molecular marker which we identified, which is KMT2D, and very hypothetic functional mechanism and outlook. So coming to the state of the art, when I started, it was not very well established setup for TLBL. So T lymphoblastic lymphoma, or TLBL, which I will refer in the rest of my talk, as T cell acute lymphoblastic leukemia, or TALL, are neoplasms of lymphoblasts committed to T cell lineage. TLBL is morphologically and immunophenotypically very similar to TALL, and they differ by the definition of bone marrow involvement. In case of TLBL, it is less than 25%. In case of TALL, if you have bone marrow infiltration more than 25%, it is T cell acute lymphoblastic leukemia.
It's often debated whether these two entities are the same malignancy or two different diseases. At the moment, it's not yet clear. Very little information in case of TL lymphoblastic lymphoma in case of molecular markers is available so far, because it's a rare disease, as well as there is paucity in clinical samples. At the treatment level, the event-free survival rates for T lymphoblastic lymphoma, which is based on TALL regimen, is quite good. It's about 85%. But in case of refractory diseases as well as relapses, it's very bad. It's about 10 to 30%. As you can see here, it's the cumulative the event-free survival rates is very low in case of relapse or refractory diseases. Unlike TALL, in case of TLBL, there is not much known at molecular, as well as clinical parameters are not well defined. For instance, sex, stage, central nervous system involvement, or age. None of these parameters associate with prognostic relevance, as well as the histological and immunophenotypic parameters, or even MRD, MDD, do not correspond to any prognosis. This is quite, it's better in case of TALL. Therefore, this does not allow to stratify the patients under different risk groups. In the last decade, therefore, the shift of attention was more towards molecular markers, to identify molecular markers that would allow us to stratify the patients under different risk groups. What do you say? Preferably when the disease is diagnosed, not when you have already a relapse. That would enable us to have risk-based treatment. So this was the main goal for our project, to identify molecular markers of clinical significance. At this point, there were two publications regarding the genetic classifier for adult TALL based on the molecular markers for RAS, PTEN, Notch1, and FBXW7. A mutational status was defined. For instance, when you have mutations for Notch1 and FBXW7 genes, those patients were having a favorable prognosis, which means they had less chances of relapse, let's say. And those patients which had mutations in the NRAS and KRAS gene, and also PTEN genomic abnormalities, which means mutations or deletions, they predicted a poor outcome. So this would allow them to have de-escalation for the low-risk patients which have mutations in Notch1 and FBXW7, and increase or intensification of treatment for the high-risk patients. In 2015, for the first time, a genetic classifier for TLBL was proposed from our group, wherein it's quite similar to the adult TALL, wherein you have 3 different risk groups. The good risk group had the patients which had mutations in Notch1 and FBXW7, but no mutations in the PTEN or in the 6q region. I will come to this molecular marker a little later. And the high risk would be the patients which are not having mutations for Notch1, which means wild type status. Wild type status for Notch1, but they have this PTEN and LOH6q mutation, so they would have the high risk. So, which means we could have basically a risk group assessment, but this was only from the German cohort. It had to be validated in an international setup. So when I joined the group, we had an international collaboration with the Italian and the French group, where we combined the data for some of the markers which were analyzed for all the cohorts, and then this classifier was validated. And the good part was only that we had 2 molecular markers like Notch1 and FBXW7, which were associated with prognosis. They associated with good prognosis, but all the other markers were not associated with any prognostic outcome. So based on this finding, a clinical study or clinical trial was designed, and which is ongoing at the moment, where for the first time we have risk-based treatment for T lymphoblastic lymphoma, where all the patients having mutations in Notch1 and FBXW7 gene will be placed under standard arm, and all the cases where you have wild type status for Notch1 and FBXW7, they would go under high risk arm, and they would have a stringent treatment. So this is an ongoing trial. So I would also bring a little notice for your attention that most of the studies till now were conducted retrospectively with patient samples, and the entire genes were not analyzed. We focused mostly on the hotspots. For example, here is an example for Notch1 gene, where we focused on the heterodimerization domain, which is the exon 26 and 27, and the PES domain, which is in the C terminus, which is the exon 34. So only these domains were analyzed because it was known in TALL that these are the hotspots, and we assume that it will be very similar in TLBL. Similarly, analysis was performed for other genes as well. So entire genes were not analyzed. And also from this meta-analysis where only 2 genes were associated with prognostic outcome, suggested us that we need to do more genome-wide data analysis and identify additional molecular markers to have prognostic impact. So for this, my project, I designed an integrated approach where we did to identify single nucleotide variants and short indels, insertions and deletions. We performed whole exome sequencing followed by targeted sequencing. To identify large chromosomal abnormalities, we performed SNP arrays, and to identify epigenetic changes, we performed methylation arrays. And we integrated these results and to identify molecular markers for prognostic relevance. So, we started with a limited cohort to perform the genome-wide analysis. As you know, it's quite expensive to do these genome-wide studies. So we did with this, we started the study with the 16 samples, out of which 10 samples were those which did not have a relapse. These patients did not have relapse, so we had the germline sample for this and a primary tumor sample for this. And the other 6 patients, they had a relapse, so we have matched germline, primary, and relapse sample. So this was the limited cohort. For whole exome sequencing, we used SureSelect XT human all exon V6 plus Cosmic Kit, and we had a high coverage whole exome sequencing. For germline, we had 100-fold. For tumor samples, we aimed for 300, but we had actual coverage between 200 and 300. Methylome arrays were performed using the Infinium methylation EPIC beat chip, which is 850K arrays. And for the SNP arrays, we use the Infinium Omni 2.5 exome. These arrays keep changing, especially the SNP arrays, and you might have a more advanced version now currently in use. So this is the first figure in the blood paper where you have the results from 3 different approaches. I will first focus on the whole exome sequencing. Here is the map plot or the mutation spectrum where we have displayed the first 50 top genes which were mutated, which were found mutated. And these genes fall under the category of NOT signaling, PI3K, RAS pathway, JAK-STAT pathway, genes related to transcription regulation, cell cycle, epigenetics, DNA repair, resistance, and others. I will focus now on whole exome sequencing for the next few minutes. This figure is the bar graph where you have the display of mutations across the 16 samples. This figure kept changing throughout the project. It was not like this. I had much higher number before, and as we changed the stringency, the figure kept changing. So the blue bars indicate mutations in the primary samples. The red part in this last 6 samples correspond to the relapse, mutations in the relapse samples. And the orange part is the overlap between the primary and relapse samples. And these could be very interesting mutations where we could already see those mutations, which if they can give a hint that there is a chance for relapse. So of course, when we started off, we had a whole exome sequencing data with about 2,000 variants, and we have to now validate this data. And of course, doing each variant validation is not very feasible. So obviously, we thought we can use a targeted sequencing approach, and we also wanted to generate a tool which would allow us to, in future, to use this as a tool to sequence large number of samples in a high throughput fashion, So to design a panel, I didn't have any template. So it was basically intuition based and common sense and knowledge based. So let's say we started off with 2,000 plus variants. We focused first only on the variants in the coding genes or coding regions. We discarded all the silent or synonymous mutation and focused on the non-synonymous mutations. And amongst the non-synonymous mutations, we chose those which had a prediction that it would have a pathogenic or a deleterious effect using some in silico tools like Provia and SIFT, and there were other tools. It was not strict though. We, if there were genes which were interesting and if the mutation was not having a damaging impact, we did include this gene. And we also chose some genes from literature which were relevant for TALL, as we know that it's very closely related malignancy. And so we had a panel about 76 to 80 genes, because the number is varying here because we had 3 different versions where 90 to 95% of the genes remained constant. Only like 5 to 6% genes we changed, where we removed some which we after the first few rounds with when we knew that it's not making sense, and if we had some interesting genes we added them. So we had the flexibility to do this. So using this panel, we validated the data on these 16 samples, and then we extended the study on a larger cohort, about 131 samples, which had 118 primary and 9 relapse samples, and some of them failed. So this is not a very nice display, but just to give an idea, when we were choosing genes, we didn't start directly with 80. We started like 200, 100, and we just reduced. And after we came down to the number to 80, and we just wanted to see whether there was any sense in these genes what we are choosing. And of course, we could see that they did make sense. They were genes belonging to some very significant pathways which were relevant for TLBL or TALL, like NOT signaling pathway, JAK-STAT pathway, PI3K, KRAS pathway, some epigenetic modifiers, cell cycle regulators, genes related to resistance to treatment, and so on. So, and here you have DNA damage repair. So, yeah, this made sense. It looked quite good, and we validated, and we have validated about 118 samples, which are primary, and 9 relapse samples. So we have the, we've mentioned the function or the pathways on the left side, the frequency of mutations, and the name of the genes on the right side. And I think you can see the pathways which are listed here. And the most frequently altered pathway was the NOT signaling pathway, followed by the PI3K and RAS pathway. And the new pathway which we characterized was the, which we also identified was the JAK-STAT pathway, and the epigenetic modifiers, which are very relevant as we go in later part of the talk. And then this data was then correlated with the clinical data to see if it had any prognostic relevance. Besides targeted sequencing, we also identified 3 additional markers, which are in the lower part of this figure. You can see left one, I can't see the lower, there is a 1 minute, this tool, okay. Left one, ABD, PTEN deletion, and loss of heterozygosity in the 6q region. And I just will briefly mention how we analyze these markers. Why I cannot. Can you hear me? Hello?
Yes, yes, we can hear you.
Oh, I am not able to go to the next slide for some reason. Why? Okay. So, yeah, so this is the marker which is called LOH6Q, which stands for loss of heterozygosity in the chromosome 6 in the Q region. And these are the deletions which we have seen for our German cohort. Just to, the method we used is MLPA, which is the short form for multiplex ligation dependent probe amplification. And another marker is to screen for deletions in the PTEN gene. And you can, again, we have indicated the deletions which we have identified for TLBL cases. And exon 7 is the hotspot both for TLBL as well as for TALL. And the panel we used is a commercial panel, SALSA MLPA 383, which is more for the TALL disease, but this can be also used for TLBL. And it's just an example how a MLPA profile looks. And this was used to look for the deletions in the PTEN gene. And you can see here the exon 1, 7, and 9. But we also have other genes here. And we also use the information for these genes. From this, we got this left one gene information as well, because we analyzed this for the entire cohort. So we could also use this data to see if there were any relevant genes for TLBL. And the third marker is called ABD. This is a bit confusing terminology. It stands for absence of biallelic deletion in the T cell receptor gamma locus. This is like when you have the absence of this deletion, it kind of gives an indication of a presence of a clonal lymphoid population. And this ABD has been shown to associate with poor outcome in the French cohort, where it was very small cohort. So we also analyzed for this marker in our cohort. So, and this results have been displayed in the lower part of this the figure. So I cannot go into details of every gene. I will just show some examples of some of the interesting candidates where the mutations are located. As I mentioned in the previous, in the beginning, that Notch1 was first analyzed only for the hotspots. And now we had the data for the entire gene. So here are the lollipop plots for the Notch1 gene, entire genes. The upper part displays mutations for TLBL, which we have identified, and the lower part displays mutations for TALL, which we got from the public database. And we use the St. Jude Pecan website to adapt this figure. And this allows us to compare TLBL and TALL just by having a glance. And you can see that the hotspots are very similar for TLBL and TALL. And the first figure are the mutations displaying in those cases which did not have a relapse. And in this second figure, you have the mutations from the cases which had a relapse. You can see that there are fewer mutations, and of course you had fewer samples too, but the lower part does not correspond to the relapse in TALL. Here is an example for FPXW7 gene, and again the data is very similar for TLBL and TALL. And I must also add here that we did identify mutations in other regions as well, but the hotspots were basically the ones which we had earlier analyzed. But we will see after we combine the data from other international cohorts whether we have to include other hotspots for TLBL. And the other genes like PTEN, which is a negative regulator of PI3K-AKT pathway, we again had mutations in the known hotspot, which is exon 7. But interestingly, we did see quite a number of mutations in other regions as well, and members from the JAK-STAT pathway, namely STAT5B and IL7R. So we could go on and on for other genes, but I'm not going to bore you with this. So we have about 80 genes screened around 118 samples, and a question now we ask is, are they of any prognostic relevance? So we correlated this data with the clinical data, and I'm just going to show some important outcome. So Notch1 was already reported to associate with good prognosis for TALL as well as for TLBL, and in our current analysis we also had the same outcome. So here is a KM plot. The dotted line indicates the cases which are mutated for Notch1 and FPXW7, and you can see the cumulative incidence of relapse was much lower for these cases which were mutated for Notch1 and FPXW7 in comparison to the wild-type cases. Notch1 was also identified as a driver gene for TLBL from our whole exome sequencing data. It's just bioinformatic analysis, so I don't have to show the details. And other markers which were analyzed previously, which is mutations in PTEN gene and loss of heterozygosity in the 6q region, both associated with higher incidence of relapses in our cohort as it was reported earlier. Interestingly, ABD, which is for the absence of biallelic deletion for the T cell receptor gamma locus, did not associate with any outcome in our cohort, unlike what was published for the French cohort. And also, surprising or intriguing outcome was for the deletions in the PTEN gene, which did not associate with any outcome, while we see that the mutations in the PTEN gene had higher incidence of relapse. This is quite baffling. We don't know at this point why it is so. Maybe we have to do some functional studies to answer this question. Now I come to the most novel part of the results from our study, which was identification of epigenetic modifier KMT2D as a marker for poor prognosis. So as you can see here in the KM plot in the figure A, you can see that those cases which are mutated for KMT2D gene, which is indicated in the red, have higher incidence of relapse in comparison to those cases which are wild type for KMT2D. And as I had shown in the previous slide, the PTEN mutations also associate with poor outcome. When we combine the cases which had mutations for KMT2D plus PTEN either or, you could identify a sub-cohort which had higher incidence of relapse in comparison to those which did not have the mutations in these two genes. What was interesting was that we saw that we selected those genes which were not having any mutations for Notch and FBXW7, but had mutations for KMT2D. So those cases where you had wild type status for Notch1 and FBXW7, if you have mutations also for KMT2D and PTEN, they had very high incidence of relapse in comparison to those genes which did not have mutation for KMT2D and PTEN. But when you look at the mutated cases for Notch1 and wild FBXW7, the mutations on KMT2D and PTEN did not have much impact. So apparently the mutational status of Notch1 and FBXW7 does have an impact on the prognostic relevance of other markers. It would be very interesting to address this at functional level. Right now, we don't have any related data from that. So very quickly, I wanted to show here some additional plots. While we see that KMT2D associates with poor outcome, a closely related gene, which is KMT2C, does not associate with any outcome, so suggesting that it is really specific to KMT2D. And Notch3, which is also a member of Notch signaling pathway, also does not associate with outcome. And some other interesting candidates also do not associate with any outcome, either because the numbers are very few or they just do not have any prognostic relevance. In a multivariate analysis, we found that only loss of heterozygosity in the 6q region and interaction term with PTEN and KMT2D had significant significance. Any other candidates did not show statistical significance in multivariate analysis. So since we identified KMT2D as a novel molecular marker for poor prognosis, I would, for the next few slides, I will just briefly talk about KMT2D. KMT2D, also referred as MLL4, belongs to the family of MLLs. These are very highly related proteins, and they have very similar structural organization. MLL4 is very closely related to MLL3, which is also called as KMT2C. In MLL4, we have 7 PhD domains, which is indicated in this blue oval bars, and FYR region, which is phenylalanine and tyrosine rich region, and a very conserved set domain in all the MLLs. Yeah, and so you have a C-terminal set domain. So what is the function of KMT2D in cancers? So MLL4 or KMT2D is required to maintain the broad H3K4 trimethyl peaks, and there are other publications where it's also involved in maintaining the H3K4 monomethyl peaks at super enhancers, thereby suggesting that it has a very important role in regulating some of the tumor suppressor genes which are regulated by the super enhancers. Loss of MLL4 in case of when you have knockout KMT2D in mice spontaneously, it induces medulloblastoma. Also, loss of function mutations in lung cancer in KMT2D is widely reported. So apparently there is quite some data about role of KMT2D in oncogenic transformations. So in case of TLBL, the mutations for KMT2D were localized in 2 clear hotspots in exon 31 and exon 48. Here in the panel A, these are mutations for all the cohort, including the relapse samples. And in the lower one, I'm just showing the mutations in the relapse cases. And you can see that in case of relapses, you have the mutations more in the C-terminus region. So it's a very large protein, which is about 600 kilodaltons. To do functional studies is not so trivial. And we decided to address the impact of these mutations in silico on the structure and function of the protein. So the ones which are indicated in red, we modeled them in silico to see if they had any impact on structure and function. This is one of the figure in our blood manuscript. It's not so good. I just wanted to bring your notice to the figure A. This is Ponda analysis, where you could find out where your mutations lie, whether they're located in the structured regions, like which is referring to the order, or non-structured regions, which is a disordered region. So if it is in structure region, your mutations, you can kind of have a hint that it might have an impact on the structure. And you can see that in our relapse cases, we had most mutations in the structured conserved C-terminal region. So we decided to look for the impact of these 3 mutations. The PHD7 domain in KMT2D was not structurally characterized, so we modeled the structure for PHD7 in silico, and we can see that the mutation which was identified in TLBL, which is arginine to proline, and this mutation indicated in red, which was identified in TALL, it lies in a loop very far from the zinc finger region. And it also has a atypical cysteine histidine motif, and it was predicted that these mutations would have an impact on the stability of the structure of this domain, and also would affect the structure on the corresponding region, which is a FYR region, which was not modeled or characterized structurally for KMT2D, and we used the structure information from the TBR gene, and we could model the structure for the FYR regions. And this mutation isoleucine to valine, this is the wild type, and this is the mutated version. So this lies in the hydrophobic pocket. When you have this mutation, it loses a methyl group, and apparently loss of this methyl group would affect the stability, and in turn would affect the structure and function of this protein. Lastly, coming to the third mutation, which is the serine at this position, which has a direct interaction with the water molecule. This is very important mutation because it is located very close to the SAM binding cleft. And here for modeling, we have used the SA, which is S-adenosyl, I think homocysteine and so on. When you have this mutation, it affects the interaction with the water molecule. It also makes this loop more rigid, and therefore affecting the interaction of the cysteine with the zinc finger, and therefore it can affect the whole structure and stability of this gene or protein, and also it could affect the catalytic function of the said domain. Of course, this is all in silico. We have to do some functional studies to confirm this, but since they are in very important regions, we could presume that they could have a functional impact, and we know from literature that mostly KMT2D has loss of function mutations. So yeah, this was the in silico data where we could address the impact of the mutations in KMT2D gene. And now I will move to the next part, which was the SNP arrays, which we had performed on the limited cohort, which was 16 samples, but when you include the relapse samples, it's about 22 samples. And this is an overview with the combined data, and you can see these numbers are corresponding to the chromosomes and the kind of percentage of alterations. And you can see that the alterations in the chromosome 9 in the 9p region were the most frequent alterations, and we had which is deletions, and we had also gain in chromosome 8 or chromosome 15. And of course, when you have a closer look, what are the genes which are in this region, that would be the most important part. We could not validate everything, but because we performed the MLP analysis for the P10 gene as well as for the 6q, we could validate some of the data for the entire cohort, and we could confirm that yes, the 9p deletions, which is already reported previously, about 60 to 70% of the cases had 9p deletions, but they do not associate with outcome. But the region which encompasses the MYC gene, we could not address, so it would be interesting to do these studies in a larger cohort to get better idea. But we could still confirm the data for some of these regions. Lastly, for the third approach, which is to look at the epigenetic changes, we performed again methylome arrays on this limited cohort, 16 samples plus 6 relapse samples. We could not get much information from this because it was just 16 samples. So the figure A, it is a NMDS, which is non-metric multidimensional scaling analysis, where you can see what are the different epigenotypes. You can see that there are 2 main clusters, and the red correspond to the germline samples. The green and blue correspond to the tumor samples, and there are clearly 2 different clusters. We do have some outliers because we do not have the same kind of tissues. We have different tissue samples. And if you look at the differential methylation pattern, obviously we have the germline samples were mostly bone marrow samples. So and tumor samples were more pleural effusion. So we expected to have a very clear differential methylation, but this is not the meaningful part. We wanted to see if there was differential methylation between the primary samples, which did not have a relapse, and those samples from patients which had a relapse, the primary samples. If they had some differential methylation, that would be very important, because then we could find some candidates to associate with prognosis. Unfortunately, the number of genes which were differentially methylated between the 2 groups were very small, and as you can see in the figure C, they were mostly hypomethylated in the samples from the primary samples which were from the patients which had a relapse, in comparison to those primary samples from the patients which did not have a relapse. Interestingly, those genes were quite interesting candidates. PPD, which is a regulator for TP53, or SOX2-OT, which regulates the SOX2 gene, and so on. So at the moment, we did not validate this data on a larger cohort, but we are extending these studies where we are comparing the methylome of T lymphoblastic lymphoma with the TALL, and also with the methylome of T cells at different stages of differentiation, mature and immature. And we got some interesting data. That's an ongoing project. Lastly, this is one slide. I just wanted to give an example how using a multidimensional approach is very important. So here is an example where you have a single nucleotide mutations in Notch1 in exon 27. These are mostly the gain of function mutations. And when we look at it, let's say the methylation profile, and we can see that NCOR2, which is also a negative regulator for Notch1, is hypermethylated, and which could result in activation Notch1 gene. And for instance, in SNP arrays, when you have a gain in the chromosome 9, which could also have a gain in if it is having the Notch1 gene there, you would have an additional copy of Notch1 gene. And all this would result in upregulation or activation of Notch1 signaling. And the approaches are very different, yeah? But if one uses only sequencing approach to look at single nucleotide variants, you would be missing out this information. So it is important to address this question using multidimensional approach. Similarly, for TP53, you might not find single nucleotide variants for the TP53 gene, but in SNP arrays, you can also see that you have sometimes the loss of one of the allele and so on. So it would be important to have a combined approach to address this question. Lastly, as you see, the project is very descriptive. We did not perform any functional studies. But we have hypothetically proposed a functional mechanism, because Notch signaling is a very well-studied signaling cascade in literature, both in terms of development as well as in case of cancer. And we could find, I came across a very nice paper or a manuscript from Frank Oswald in Nucleic Acid Research from 2016, where they show that regulation of Notch target, expression of Notch target genes is a very tightly regulated process, where you have 2 regulatory complexes. There's a very dynamic interplay or fine-tuning between these 2 complexes. One is a coactivator KMT2D complex, and the other one is a repressive NCOR complex. And both these complexes compete to bind at the same site which is a Spock domain of a sharp protein, and they could result in either activation or repression of Notch target genes. So I'm not going into much detail, just to give a scenario where, for instance, when you have hypermethylation, as we see in our cohort for NCOR2, you would not have expression of NCOR2, and probably this would lead to an unstable repressive complex, allowing the co-activator complex to bind, and then this would more support the activation of Notch target genes. In the other scenario, where you have loss of function mutations in KMT2D gene, which is a very important member of this activator complex, or if you would have a phosphorylation of KMT2D gene, which is a downstream target of PI3K-AKT signaling cascade, in both scenarios, you would have loss of function of KMT2D, and maybe this would result in an unstable co-activator complex, resulting in the repressor complex to bind and resulting in repression of Notch target genes. And as we know that KMT2D regulates also the tumor suppressor genes, it would result in repression of tumor suppressors. So I think this interplay with this has to be a very finely balanced system. When you have the derailment of this function, and you would have different malignancy depending in which tissues you have this derailment. And I think this could be one of the mechanism in TLBL as well. We do not know at the moment, but yeah, this was one of the hypotheses we made. With this, I come to the end of my talk. To conclude, this is the first systematic genome-wide comprehensive integrated analysis for pediatric TLBL. Notch1 is identified as a driver gene for TLBL, and we identified Notch and PI3K-AKT signaling axis as well as alterations in the cell cycle regulators as the core oncogenic program for TLBL. We did identify other pathways like JAK-STAT and RAS signaling pathways and PI3K-AKT pathways, and most of these pathways are activated in case of TLBL as well as in TALL. Notch1 and FBXW7 mutation status has significant impact on prognosis of other molecular markers as well. And mutations in KMT2D were identified as novel molecular marker associated with poor prognosis. And the findings from this study present a high translational potential to improve the treatment outcome of pediatric TLBL because these findings are being prospectively validated in the ongoing trial for TLBL, which is LBL 2018, where we have designed an updated version of a targeted panel, which we used part of it from the data I presented now, and then we removed some of those genes which we saw that they were less frequently mutated. We added new genes, and we have an updated panel, and this is one of the secondary endpoint for the clinical trial, and we are able to sequence a large number of samples because this clinical trial is among 22 European countries and more countries participating. So this will allow us to validate on one hand and also identify novel molecular markers in near future. And some of the cases, like maybe I think 2 or so, did not feature any mutations in the genes which we had in the targeted panel, which suggests that we need to perform more genome-wide studies on additional cases or larger cohort of samples to identify additional markers. In the lab, another aspect we are looking is clonal evolution in TLBL, as we are having the access to tumor samples at different time points of the disease. And as I said, we are comparing the TLBL methylome with methylome of TALL, as well as T cells from different stages of differentiation. And finally, we have also started doing some functional studies for some of the key pathways. Currently, we are looking at the mutations we identified in KRAS, and we have a hint that they have an activation impact. Yeah, so thank you for your attention, and I'm ready to take questions.
Thank you very much, Dr. Tasneem, for your very insightful talk on TLBL and TALL. Can we open the floor for some questions? Okay, let me start. Right, Dr. Tasneem, in the first few slides, you mentioned that you have chose exon 26, 27, and 34.
Yeah.
And I saw that the chart there shows that you have different types of mutation, the missense, deletion, insertion, and so on.
Yeah. Yeah.
So on what sense did you actually ignore the others and only took the, if I'm not mistaken, the synonymous mutation?
No, synonymous are otherwise called silent mutations. We don't take, I mean, initially what do you say, the study was very Sanger sequence based, they just looked for mutations all the mutated cases were included. In German cohort, I think everything was included, and they saw that mutated cases corresponded to good prognosis. But later on, now in the clinical trial, we focus more on only non-synonymous mutations. We don't consider the silent mutations. So
I mean, my question was, on what basis you found out that the other mutations actually gave a damaging impact to the prognosis or to the disease progression and so on?
So for Notch1, it has been well studied for TALL. They have shown that most of these mutations have gain of function. We do not take each mutation per se. Yeah, you just for when you want to say, if, for example, I give the example for this trial. We just look for whether it is mutated for Notch1 and FBXW7. We are only focusing on the specific hotspots because these were the hotspots which were analyzed in 3 different international cohorts. So we are not going variant by variant. You just look whether they are missense or non-synonymous mutations, and then you consider them as mutated and you put them under standard risk.
Okay.
So at functional level, we are not addressing because it's known in literature that they are mostly gain of function mutations.
So we have got a question from the chat box here.
Yeah.
You find KMT2D mutation is a marker for poor diagnosis. How are you going to use this to use this outcome for enhanced therapy, or how you translate your finding into clinical setting?
Yeah. So, for instance, as I said, now for the first time we have the clinical trial where we know that Notch1 and FBXW7 mutated cases are coming under good risk or standard risk arm. Yeah, but we do not have any molecular marker to identify the poor risk arm, which is the most important part. And now from our study, we know that KMT2D associates with high risk or poor risk. Yeah. So what we do is now we have this in our panel, in the targeted panel for TLBL, which is used in the clinical trial. So we are able to prospectively validate our findings in this ongoing clinical trial. So we have 22 plus countries participating. So we will have huge number of samples which will be analyzed. And then we will correlate the data to the clinical outcome. And if we could confirm that indeed what we found in our German cohort is really good for other cohorts as well, then you could have another clinical trial where you could have a escalated treatment or a stringent treatment for the high risk group, because you're able to identify when you have a patient coming, you can do the profiling, and if it is mutated for KMT2D, you know that this comes under high risk and it can be subjected to intensification. But this is the plan. It's like an idea, but at the moment it's still being validated.
Right, another question from Dr. Hyo, who is our IMMB director. Thank you for the interesting presentation. Would you think the results would be different if you were using Asian population?
Actually, this is one of my criticism as well, because most of this clinical trial is mostly European, or you have America, Europe, and I find it quite difficult to not have a representation of the Asian population. I don't imagine it will be different, but in our study we have included even China, so this would be interesting to see how it would be. At the moment, I cannot say if it would be different, but we have this question as well, and we are looking forward how it will be, because we have Hong Kong also in the trial, and yeah, this is awesome. Yeah.
Any other questions? Do we have any questions? How do you actually identify if that particular mutation comes from a primary tumor or a relapse? Are you looking at expression levels?
No. So when you have a patient coming, when you have the patient, when it is diagnosed, then you get a the diagnosis. When you get a tumor sample, it's a primary sample.
Yep.
And then the patient is treated and goes under remission. When the patient has a relapse, in case of TLBL, normally I think in the first year itself, you might have the relapse. And when you have a relapse, then you collect the sample from the relapse condition. So you already know that it's from relapse condition. So, And for germline, you take any other tissue which does not have the somatic mutation. So depending on when you collect, you would have either primary or relapse.
It's already a known relapse case.
Yes,
Yeah.
yes.
Not that you predict relapse.
No. But we based, so the idea is when you have a case coming, like primary case, and if you would have a mutational profile, and we knowing when we do this, let's say if I would get a patient sample, and if I check for Notch1, FTXW7, and KMT2D, and if I have a mutation KMT2D, I could predict that this might have a relapse later,
Right.
But we do not predict it whether that the sample is relapse. We can predict. That's the idea of having this prognostic marker.
Okay. Thank you. Any questions from the floor?
I'm sorry about the confusion with this time difference.
Okay.
I don't know. I thought 9 o'clock means 9 o'clock. I did not.
We understand your time difference there.
Yeah, it changed just this weekend. I think it was yesterday. Yeah.
Right. Say if KMT2D is actually a marker for diagnosis of LABL and LTBL and LTALL, right?
Yeah. No, we are only talking about TLBL. We don't know about TALL. Okay. We haven't looked in TALL.
But the gene itself is actually controlling the tumor cell regression, right? It suppresses the tumor suppressor genes.
Yeah, it regulates several genes and also tumor suppressors.
So
it has
the expression or the mutation of this particular gene, would it suggest of having any other kind of tumor other than this?
Yeah, for instance, you have in medulloblastoma, loss of function in KMT2D. In lung cancer, you have loss of function mutation. So it can be in different malignancies. Whether it is prognostically relevant, we don't know. But for this, you need to do a study on a large cohort. But yes, they have shown that there are loss of function mutations in other cancers as well.
Right. Okay. Thank you. Can we have another 1 or 2 questions from the floor? If we have.
Do I have to read or you are reading?
No, I'm going through. There's no questions yet. Right, this is not a question. I'm just asking you, Dr. Tasneem, do you have any collaboration in this region in Malaysia?
No. Malaysia, not. It was, I think, the Chinese representation was from Hong Kong. It was 22 European countries, and we have also some other regions, but not much of Asia. I don't know why in such studies there is not. I think because they want uniformity in the way the patients are treated or diagnosed and so on. So I don't know, maybe in future it can be different, but China is making a lot of efforts to be a part of such studies. Yeah.
So you're definitely open for any kind of collaboration.
That's a question for my boss. I mean, it's a big consortium network, and I think there are sometimes invited from Japan and so on. There are people participating from Japan, but somehow they do not take part in the trial. But they take part in all the conferences and discussions, but they are not part of the trial.
Right. Thank you. I think there's no questions from the floor, and I think I'll have one more question. How do you actually do functional studies on this particular mutation that you have identified? In terms of structure, you have showed, but how do you show that they are actually functionally related or correlated to the disease?
Yeah. So for the disease, I think you would have to first of all do something in the cell lines or in the mouse model, where you could express this mutated version and see what happens. And of course, you have to do then tissue-specific expression, maybe in T cells or lymphocytes and so on. And we know that the SET domain is important for the histone methyltransferase activity.
Yeah.
And this can be really addressed, whether you have the methylation being affected when you have this mutation. and one can do in vitro as well as in vivo studies for this. I mean, for the set domain, and when you have the mutation, whether the expression is changing or whether the, I mean, you can, depending on the mouse model or in vitro, you can address different questions. It's for the C-terminus, because it's in the structured region, it is quite, what do you say, easy to plan, but not easy to perform. It's a large protein, but of course, it would be a very interesting project to address at functional level.
So the established animal model is mouse, is it, for the disease?
In our lab, you know, when we started, it's a very clinical setup. I mean, it's a clinical lab where you have a study group, and part of it is functional. They haven't had, and they haven't entered into the animal models yet. Their plan is to do it in the zebrafish, but I think most models are more closer to the what do you say, the mammalian system. But I think quite a lot of work can be done with the zebrafish model as well. But at the moment, it has not been established. We are using cell culture, and there are not many cell lines for TLB, unfortunately, as well. Yeah.
Thank you. Any more questions? Anybody has got one last question before we end the session? Anything from the floor? I think there's none. Thank you so much, Dr. Tasneem. We appreciate that you have allocated your time in spite of the zone difference. Okay, I believe it's probably early in the morning there. We really appreciate that you joined us for this webinar series. Hope to see you again.
Yeah, that was my pleasure. I'm thankful that you invited me, and I think I should thank Tang also, who introduced me to you guys. So it was very nice for me to have an opportunity to present this work.
Thank you very much again. Hope to see you again or meet you again in our next webinar series.
Okay, I hope you had you could understand my talk.
Yes, definitely.
Thank you.
Thank you, everybody.
Thank you, everybody.
Thank you, Dr. Tasneem.
Thank you.
Thank you.
Thank you.
Thanks, Prof. Tang also. Thank you.
Thank you, Dr. Tasneem.
Thank you. Thank you.
Thank you, Tasneem.